Thanks, Frederic, for the invitation. It's very nice to be here, and I just had a tour about this amazing center and platforms, which gives me many inspirations to take back to Glasgow. I'm in two centers, the CCNI and the ICE, and we will restructure them into one big center. So that was very good. I also enjoyed the weekend. I went to Zermatt and did some hill walking, and I had some beautiful. Actually, this is when we moved the presentation. I missed this one slide, so otherwise I would have shown in the end me in the mountains. So I'm just very happy that I had the opportunity to also enjoy the Alps, where I'm actually from. I'm pretty close to Lake Constance, but now for 18 years in Scotland. And so let's start. What I want to talk about is layers and I will mostly talk about primary visual cortex, layer fMRI, and the idea that in the primary visual cortex you have internal models that are predicting the outside environment. And we compare layer-specific fMRI with rodent and monkey research, which was possible through the Human Brain Project. We extended our collaborations with doing the same experiment in rodents and monkeys, and then also modeled around this, and we continue to do that work, hopefully going forward. But so you get the snapshot. Frederic said I gave a presentation at ABIM 2 years ago, but I had, of course, to put in new stuff. So I will start with a different focus, and the focus is the influence of experience on the predictions in early visual cortex. So this is the question: how does memory shape your predictions? Very important, and it's a bit of a detour or an evolution of my research so far. I'm going to start with 3 experiments in which we show how memory traces enrich the predictions in early visual cortex. Let's remind ourselves of the challenges the humans face in our environment and the visual system. On a sunny day in Amsterdam, that is a typical street view, and you can admire how people navigate without bumping into each other. There's lots of prediction going on, and there's also some occlusion going on. So you see there's a truck driving through, and you know that there are bikes behind the truck, and you can still, or the van, and you can still anticipate where people are going. You read their intention, and you don't trip over. Quite amazing to see. So that's the visual system helping us to survive. And we understand the brain to have this organizing principle in cortical layers, which we believe is the place at which 2 processing streams come together. I pick out the visual cortex for technical reasons, historical reasons, because I'm trained, and all the kind of experiments are based on retinotopic assumptions. But the principles that we derive from here are probably true throughout cortex. The idea is that the blue stream, the sensory input, feeds into visual cortex. So of course V1 is the first cortical stage at which the processing is happening. And then you use the top-down predictions, all your experience, to kind of interact with expectations and amplify or disamplify information that fits to that or is a surprise. So that's in a nutshell. The pyramidal cells, the big one is a layer 5 pyramidal cells, the small ones are layer 2, 3, are the most common cell types, and they have some important principal features that are essential of combining these information streams. If we take a layer 5 pyramidal cell, it has such a long apical connection that if it would work as an integrate and fire neuron, the 10,000 synapses that are in the apical dendrites, so in the superficial layer, would never reach the soma and it wouldn't trigger an action potential. So integrate and fire, although it's the dominant concept, can't be working. So Matthew Larkham describes, for example, in this TINS paper in 2013 already, the idea of two-compartment neurons, which is central to many ideas that we are having here. How that works, in a nutshell, is you have all the feed-forward information coming close to the soma of a layer 5 cell. The feedback comes to the superficial layers. The feed-forward triggers an action potential, and a back-propagating action potential reads out what was known about the situation to the apical dendrites. If there is a certain constellation or depolarization, then the whole cell starts to burst. So it's a different firing pattern dependent on the integration of cortical feedback and feed-forward. So that was the theory that we also set out to test. Or that's the thinking behind how it could work, that feed-forward streams and cortical feedback streams are read out and interfere with one another. So you can amplify, disamplify them depending on coexistence of these two streams. We know from anatomical connections, Kennedy lab, for example, that in humans and monkeys in their study, there are two dominant cortical feedback streams, one to the superficial layers, one to layer 1, and then to layer 2, 3, and to the deep layers. So what's shown in pink are the potential feedback layers, and the feed-forward goes to layer 4 and the mid layers. So to investigate my original situation, we need to control it a little bit better and to slow it down. So we just take a still frame of natural scenes. So that's many of the experiments are based on scenes like this, just a still frame. And so we have, in this original study, we have 24 different images from different categories: trees, streets, offices, houses, and so on. And then we have 4 different examples. And we always occlude one part of the visual stimulus, the lower right. And in that part, we do the recording. So this is a part of visual cortex, V1, layer specifically. So recorded with 0.8 millimeter resolution. And the rainbow colors show the different depth values of V1. But it is the position that corresponds to the occluded areas. So the feed-forward input is always the same and non-informative. And the original study in 2015, we then found, if we do decoding, that there's an increased information to the outer layers, so the red and the yellow. And the outer layers, we get information about the different scenes. In that original case, we only had 3 visual images. In this escalation, we have 24 different scenes. And what allowed us in this study was to do different kind of modeling. So we tested H-max models of different hierarchy, depth model, and noise level Weilball models. That's work done by Tyler Morgan. And we came to the conclusion, so this is V2, but the GIST model was the best explanation of the occluded area. GIST model here means not the GIST model was derived from behavior. We asked 50 subjects to do a line drawing of information that they expect behind these scenes. So you would be able to do that yourself now. If you have like 30 seconds to sketch out a certain line drawing, if all of you have different internal models, then it would come out homogeneously gray. But you would probably draw something like this. So that's what our subjects did. And if we use this line drawing and make a GIST model, that's a good description of the activity that we find in these occluded areas. It's better than the original images. So if we would take the full image and do a GIST model from that, it wouldn't be as good because it's richer than your prediction. So that's the idea. And Paolo Papale in Peter Wolfsamer and Matthew Self's lab used 2 trained monkeys that were trying to fixate and had already electrodes and then penetrated electrodes into V1 while presenting the same visual scenes, 24 different images. Here you see the beaches, for example, and the receptive fields are recorded in this occluded area, like always. So they're not responding, the core receptive field is in the occluded area, and you see the responses here in blue when you show the full image. That's the typical response you see, and in the occluded area, when it's occluded, so to the right, you see this little bump, and it has a 40 milliseconds kind of an offset, which is consistent with the feedback, and it comes with the same delay of any place within the occluded area, which speaks for cortical feedback and not for lateral interaction. A question is often raised. We have various arguments why we think it's cortical feedback and not lateral interaction, or it could be a bit of both. But so that's the first piece of information, and the second piece of information from this study is that if we use, so what you see, this little inlay is now representational similarity matrix of our human data. So those are all the 24 different scenes in the column and 24 in the rows. And you see how the response in the occluded area is correlated. So that's the human correlation matrix. And in the monkeys, we can use, because it's electric recording, so we can create these dissimilarity matrices every few milliseconds and compare that to the human and correlate these two matrices. And you can see here that after 180 milliseconds, they start to correlate and stay correlated, which means the idea, since we know that this is correlated to the line drawing, you can assume that the monkeys have something like a sketch of the occluded area as well in the primary visual cortex. But we don't know exactly which layer. So the monkey electrophysiology, it's not so easy to, you need to guesstimate where it is. It could be deeper than the superficial layer, but we get in the next study. The mouse not working. We went to rodents and used calcium imaging, and now you see the marked cells from which the activity is recorded. And in this, so there's two ways of doing this, with wide field imaging or with 2-photon calcium imaging, and both work well in the superficial layer. So we're looking into layer 2-3, and so there you see individual cells that are responding to either presenting the whole scene again, or the occluded version of it. There's a small variation. They are presented slightly differently, and the mice need to be trained and learn that visual stimuli are relevant. But after they have done this, you can see that this is a comparison of human data to mouse data. This is about the information contained in the occluded area. And here we do cross-correlation, cross-classification from non-occluded to occluded, so non-occluded to non-occluded. So that means feed-forward images that are classified, occluded to occluded, so it's both wide and then cross-classified from the one to the other. And you can appreciate that the profile between human and mice are relatively well compared. And so they capture some of the same features in these layer 2-3 cells in rodents. And of course, I'm quite pleased after I started neuroscience in the lab of Wolf Singer in Frankfurt, the Max Planck Institute. So we were doing brain imaging alongside the gold standard of doing monkey and cat recording, and my colleagues always teased me saying, so why do you do this expensive fMRI? You basically rediscover stuff that we already know. And so this is one time that we turned around the paradigm that we developed in fMRI, that we came to layer-specific hypothesis, then put back to the neuroscience people, and they found something they didn't expect. And it was quite interesting from the first meetings we had during the Human Brain Project. The typical visual neuroscientists working in rodents, they're like, but can we do gratings, or can we, why do you want to do, and why is it occluded, then it's not going to respond. So it was really, they hesitated that it would work, and it worked with the layers. So that's nail-biting moment that I've been waiting for 20 years to then get something replicated across species, across method, and so I'm quite pleased about this. But now we learn something very new, and which inspires the next row of experiments. And that is, the rodents have seen a subset of images for 2 weeks again and again every day. They go for half an hour into the maze, see the images, they get recorded during the stimulation, they get feedback for just looking at the images. So here's the same way of showing the data like in the monkeys. So it's an occluded area. If you show feed-forward result, that's the black line. Oops, sorry. And then you have this little hump for the feedback activity specific to the images. But that's after 2 weeks of learning. You see the summed response of those cells are responding now when it's occluded, the red line, almost as strong as the black line. So repeating the same stimulus again and again reduces the response, which we know it's called adaptation traditionally or so. This is the response on the right for the overtrained stimuli, and on the left is, which I haven't said yet, is after 2 weeks of training, they introduced a few more of the images of our data set. So after they have learned they need to take attention, new stimuli are included, and those are the same stimuli, so they don't make a lot of sense for the mice. So you have a skyline that they have probably never seen, and you have beaches from above, and offices which they haven't never been to. But these images make sense in the sense that we can look at pictures that we have never been at that place. We still understand the gist of the scenes. And so that's what the rodents are doing on the left. But on the right, those are images that they have seen for 2 weeks again and again. And what's very surprising is that the occluded response now is really strong as the non-occluded. So now the big question is, what happens of the red? Why does it increase? And what can we see in humans? And this is now news. Yeah, okay, this is just the same image, it's a little bit bigger in case you couldn't see it. But this is the time point correlation that I presented. So the representation of the similarity, and then after 120 milliseconds it starts to correlate. And this is the setup for the mice. I summarized that already. So here's the one bit of new information which is quite exciting, which we also didn't expect. So now you have this response. So the red are the cells that have learned over the 2 weeks to fill in the information. They know the stimulus and suddenly it's occluded, so they know exactly what to expect behind it. And the first way of thinking about it is you have these prediction neurons that kind of fill in now with top-down drive what should be there, like the mental line drawing as we described earlier. So you have the monkeys that make the line drawing, or the humans that make the line drawing, and those cells are now representing what should be there, the line that isn't shown. If that would be the case, then these cells would respond along the diagonal. So they would respond in the situation that the stimulus is shown, that is here on the right, and when it is occluded. And in this scatter plot you see there's hardly any along the diagonal. They fall into two clusters. One cells that are only responding to the non-occluded or to the occluded. So they have learned, so this population is increasing, so it's a new population that has learned what's there. And so how to conceptualize this? One way of thinking, which might in the end not be the one we favor, but one way of thinking about it, and Chris Keller and so on had predicted this, is you have negative prediction error cells. So what that means is you have a top-down signal that says you are expected, so I expect now there to be a line, but it's not there. And so that's the negative prediction error, and now I'm firing. So it's something that's very much expected because you know it and is not shown. A positive prediction error is you show something and you're surprised by it. So that's on the left. You show something and you're surprised, a positive prediction error. Or you leave out something that you expect, negative prediction error. And they fall in two different categories. So it's two different cells in layer 2, 3. We don't know what separates them, but there are two different populations. One's that learn about the environment, and the other one that respond to surprise. So positive and negative prediction errors in two different categories. So now we want to see if we can find memory traces in humans. So the challenge is we present a, let's say, a new scene, and you have never seen this scene, but you have some expectation what is behind the occluder. And we want to investigate if we can enrich this kind of memory trace. So, for example, if you have been experienced, so there are three forms of information here. One is just driven by the surround, and then one by you filling it in, or a third one by you knowing what's behind there because you have seen it before, so memory enriched. And in order to get to this memory enrichment, we use Mooney images. So this is a context image which you don't make a lot of sense of. If you haven't seen it before, it's not enriched, it's just black and white. How many recognize that? You want to take a guess what it is? Anyone? Okay, so have you seen it before?
No.
Okay, you're correct, it's a waterfall. So we have different streams, and we make them out of different categories just to keep it on side. So the second one is a face, and the third one I don't remember. So three different categories. In our fMRI run 1 and 2, we just show black and white. Then during the anatomy, we show the grayscale versions. Now you can see very nicely the waterfall, and you can recognize the face even though you just see a little bit of it. This is an animal in the end. And so now they get familiarized during the anatomy 10 minutes, and then comes run 3 and 4, where we show again black and white. And we always occlude the right lower visual field, and we do retinotopic mapping as always. So we use retinotopic mapping to separate V1, V2, V3. These are the patches that are specific to the occluded area only. And you can see the dissimilarity matrices in the response of the occluded area, so to the gray patch, before and after the memory enhancement. And so, yeah, you can see how they become more dissimilar after the anatomy, after you have some experience and understand there's waterfall and faces and so on. So that's what we think. Feedforwardly, you had the same identical input, but now it's enriched with knowing more about the scene. So that was the first memory experiment. So after learning of the meaning of the context, the top-down projected information in the occluded part becomes more meaningful. Now the second one is the question knowing about, so this is like making the context more meaningful, so recognizing the building and the park. Now the question is, can we implant memory traces? Like in this case, there's actually a pond. You don't know that. The context didn't give you any idea that there's a pond. But once you have seen the full image, you might remember that pond. And that trace of memory might influence the top-down stream and fill in the missing information. In order to look into this, Javier Ortiz Tudela, who was in the lab of Yi Li in Frankfurt, conducted this experiment in which they had 4 different rooms and with 16 different objects. So kitchen, living room, bathroom, and so on. And in these contexts, there were items placed, and on day 1, the subjects went through the items and saw them again and again. So you would have, let's say, a kitchen with a bathtub or a living room with a bathtub, and you would remember that that scene had that item in it. And then on the next day, they were scanned with the occluded version of that scene, and we were then looking whether there's evidence for the bathtub that they have learned on day 1 behind the occluded area. Now, a few advantages of this experiment is that all the rooms had only one line that go into the occluded area. So, you know, when I say the mental line drawing, We wanted to keep that at the minimum. If you do a mental line drawing, you would always just have this one line. Everything else was kept the same. But you now had a memory trace of these objects, and which is the episodic memory experiment. And we could find traces of that in V1, V2, and LOC. Now, it's not decoding. In most of the other experiments I've done decoding. This is a correlation method based on, I'm just wondering if I have a method. Yes, so I'm going to show it to here. So you have the dissimilarity matrix when the room changes and the object changes. That gives you one dissimilarity. And then you have a dissimilarity when the object in the occluder is the same, but the room changes. So you have a little bit of an increase of similarity if your memory trace works. And that's what we are showing here. So the increase of similarity because you're memorizing the same object, so the bathtub, is what we see above the zero line. And so episodic memory, it works in these areas, and semantic means what we have done here is we didn't train the subjects on day one, and we just gave them the different rooms and looked for objects that are semantically related. So the bathtub should fit into the bathroom, but that by itself didn't trigger a response or an increase in similarity or decrease in dissimilarity in V1 and V2, but in LOC. So it needs the memory trace, remembering that it was there, to make that work all the way down in the hierarchy. So that's the second example of how memory enriches the information in visual cortex. Episodic memory traces of objects behind the occluder can be traced on the next day, shown here as an object reinstatement correlation of multivoxel pattern analysis. And then comes the third part, which Matthew Larcombe published this experiment. He talks about a memory switch in the rodent experiment. He stimulated with electrical stimulation and a whisker response. And after 8 or 10 repetitions, the rodent had learned it, and they could disconnect it with the hippocampus, and this memory trace worked in cortex. So you build up this kind of memory trace for which you need the hippocampus, and afterwards you have the quality in cortex. So that's the thing that we were kind of thinking, can we relate that? So here we were looking to a effect for one single image, or 8 times repeated image, or very new images. So we had to adjust now our experiments. So far we presented our stimuli again and again and again, and then this whole logic doesn't work. So here, for the first time, we switched now to category-specific classification. So the images are either cities or offices, and we have a pre-training session in which we show some images once and some other images 7 times. And then we go to the scanner and have the one image shown again. So it's not novel, it's a second time, or 8 time, or novel. So those are the images, and they're either cities or offices. And so the result here is now for category classification. First of all, we weren't quite sure if it works, but it works. And now it works only for very familiar, and so we are categorizing in the occluded area. In the non-occluded area, you have lots of feed-forward information, and it's probably not working. You wouldn't be able to get the memory trace. And so in this occluded area, we have no information of the novel, but it needs, or one time shown. Maybe it's familiar is a bit of a old slide, not a good term. So familiar means here one time presented or very familiar. So it needs to be presented 7 times, and then you find a memory trace. And this is layer-specific fMRI again, and you find this memory trace in deep layers. So it's fascinating to show this, but we're surprised to find this in deep layers. So if you remember, in the other studies we show this in the superficial layers. But of course there are a few things different. We have a very specific prediction in the one case. In this case you show the same image, 3 images, many times, and you know exactly what to expect. And in this case we try to classify across all different cities and across all different offices. And this classification only works once you have seen the offices and the city of that specific type, but it's not making a very precise line prediction. So just to make sure, we rerun the experiment having a data set of our original case and of the novel case together. So this is the 2015 paper where we had 3 images. We repeated them 32 times in the same way we did it originally. And then we have this categorical classification, familiar and very familiar stimuli. And we replicated this crossover. So for the overtrained images, you find this in the superficial layers, the lines. And in the categorical classification, you find it in the deep layers, only for the familiar one. And so this is very new data. We're just writing up the manuscript, and if you have comments about this, I'm pleased to hear this. We're quite excited about it for various reasons. One, there's some theoretical belief about layer 6 cells, that there's some rodent work from Matthew Larkham again that actually predicted something like this, an abstract categorical lookup table that associates across cortex. And so it might be related to that. And of course, it's also in the same subject, same session, finding a feedback to the superficial layer and finding a feedback to the deep layers is intriguing and interesting, because it shows that our results are not biased by, for example, the amplitude increase of EPI responses that you are predicted to get between the layers. We might have that, but the classification is on top of that and shows this variation. So this is the third conclusion. So contextual feedback for images. Contextual feedback for images, the mental line drawing kind of feedback goes to the superficial layers, but the abstract categorization of cities versus offices goes into the deep layers, but only for familiar images. That's where we are right now with that conclusion. And we have this, we kicked off a debate about how much cognition is there in the feedback of V1. So that was CCNI, adversarial, generative adversarial collaboration, started last year in Boston. It's fascinating. There are colleagues like Tony Mofshon and Peter Rosema on the other end, and like 8 other colleagues, Peter Vetter, for example, and we are debating about this. We look at the same data, and Peter has been challenged, for example, that it was claimed that some of the feedback results that he's seeing, memory results, for example, in monkeys, might be related to small eye movements. Then that couldn't be fully debated. And then we generated an experiment. We said, okay, how would you experimentally approach it? And then we designed one. And then they had monkeys recorded. They recorded the data and gave it back to the community. And even though we pre-designed the experiment and we look at the same data, we made a cloud of opinions, after a year of debating, and we still end up in 2 different clouds. And I thought that's not the case in science. You don't end up in 2 different, but you need some tools to question your beliefs and so on, to get to that point. Usually if you give a talk, you think like, okay, now I've convinced them, right? And you go out and you feel like, okay, I communicated this concept. But what you're not aware is that, half of them are stuck in another belief system and don't trust your data and so on. And that continued for a whole year, and it's fascinating to see that you can be in different echo chambers or so. So I'm on the other extreme, trying to show, to which, yeah, I'm going to say in a minute. So we find all kinds of evidence, and I showed you now the memory experiments that are aligned here. I'll show you maybe two more and wrap up. That there is some cognitive feedback to be won, and I think it's quite helpful because we have a rich, difficult task to do in the environment, to navigate, to remind you to the navigation of the bikes. And sometimes traditional neuroscience concepts run out of complex cells that come way later, where you solve and coordinate and so on. So I think there's a 70% chance that any area is connected to any other area from Kennedy Lab's data. So there is some connectivity, and in my opinion, it contributes at an early stage to contextualize your input with your output, and it makes sense, but it doesn't please our engineering mind, which tries to keep it simple and build up a more complex case. To which Tony Morfson would say, well, we need to agree that nothing that comes from human brain imaging is trustworthy. And he also rejects the rodent data, because rodent data and human data are so different that we can't draw any inferences between that. So I tried with that talk to steer evidence against that perspective, to show that there's cognitive feedback to early visual cortex, and that rodent data is actually quite meaningful in adding to our interpretation and understanding. At least it was meaningful for us, because it motivated us to go back to look at human data and the memory trace. Yeah, so I promised with the original title 2 different feedback streams, and I'm just wanting to highlight one other case in which we have the 2 feedback streams, and that is a case in which we compared imagery versus illusory perception. That's the Johanna Bergmann study. Here we have this illusion of a neon color spreading, of either being red or green, and we instructed the subjects to imagine a red or a green disk. And then we used multivoxel pattern classifier classification in these non-stimulated areas again. So we mapped out this area here, this area. And what we found is that for the illusory contour, that's the blue line, you can classify in the superficial layers again. So you have a concrete feedback to the superficial layers showing you what to expect. And when you are instructed to imagine a green or red disk, classifying whether it's green or red, so the content, so it's not just an overall attentional shift, but it's the content, whether it's green or red, can be classified in the deep layers. And so that's another example in which superficial and deep layers have a different purpose. Now, you know, there are more questions. If the 2 data sets are, the later one is very new, but you know, it could there be a link with categorical? Is green and red the different category? Is there more an abstract representation and so on? So we need to think more about how these, we need to publish them first and then think about more how that could be linked. As another reference, Jeff Hawkins has written a book called The Thousand Brains. The first one, 2002, was on intelligence. I don't know if you know Jeff Hawkins. He programmed the palm, became a billionaire. Originally, he wasn't allowed in Stanford for the graduate school, but then he bought, after being a billionaire, he bought a neuroscience institute to do the neuroscience, and he's leading a company called Numenta. And he has written this book, and on intelligence inspired me to think about predictive brain. It's quite inspiring. The second book now, called 1,000 Brains, has the hypothesis that in cortex you do the same thing as the hippocampus is doing in his mind. So hippocampus has been ingenious invention for evolution, and the cortex just copied some essence of that, made it a little bit simpler, and that's what you find in the column. In the column, so he claims, you find individual images. So first time you come to a place, you get associated in the superficial layers, and the more and more you know that place, you build a more abstract representation of that. And the more abstract is in the deeper layers, let's say. He's jumping a little bit over the reference. He's not the, yeah. So it could be that those two studies come together, that in the deep layers you have a more abstract representation, and in the superficial layers a more pictorial representation. And maybe that's a way of contextualizing what we have shown so far. Yeah, and in the interest of time, I think it's good to wrap up now. So we now I'm going to show you one more study, and that's the auditory. I promised that. So we blindfold our subjects, and we present them these different tones, 3 different categories. So let's work with Petra Feta. And the third one is a cocktail party. And we used this experiment, we used retinotopic mapping earlier, V1, retinotopic mapping, V1, V2, V3. And you'll find decoding in the periphery of V1, V2 and in normal subjects. Then we replicated this experiment in blind subjects. So they are congenitally blind. We needed to align the cortex to the retinotopic, to the anatomy, to the retinotopic in sighted subjects, and then superimpose it. And with that strategy, you can get early visual cortex, V1, V2, V3, responding to the auditory, but more so in the periphery. So we think of that as you have some original connection of small number to the outer periphery. There's some monkey connectivity evidence for that to be true. And then if you're congenitally blind, this proliferates and these connections grow and get stronger, and so you have this decodability of auditory information. And now we use the same experiment in aphantasic subjects. Aphantasic subjects came to the lab. They report themselves that they don't have visual pictorial images, and we tested them in an objective way. So we have a binocular stimuli and then see whether we can bias their preference by an imagery task, and so that's in a way you can. So this group are hypophantasic or aphantasic subjects, and you can see that for the sounds given, presented in the scanner, you don't have the classification in early visual cortex. You have a little bit in the periphery of V1, but it's massively reduced. So we think that this kind of auditory top-down feedback stream to V1 is reduced. Okay, so let me wrap up. We started with the Matthew Larkham idea that the layer 5 pyramidal cells are integrating feed-forward and feedback streams. So in that paper he proposed that you have the motion and the color of different in different streams connecting to V1 as the context information to the layer 5 pyramidal cells, which are then integrated. We diverted a little bit from the model to find more rich layer 2-3 cells that are included in this context as positive and negative prediction errors that enrich this contextual processing and make this dependent on your experience, dependent on the images you have seen and the context. And then while this automatic thing is ongoing, you also have a second level in which you do cognition imagery, like I showed in the last image with the red or green. You can imagine something, and you do this in parallel, possibly separated in different layers of primary visual cortex. And with this, I want to thank you for your attention, and my lab and collaborators, especially through the Human Brain Project, Koen, who did the mice recording that I talked a lot about, in Christian Liefeld's lab, Paolo, who did the monkey recording. In my lab, it's Lucy Petro, who's been working in pretty much all of the projects that I've presented in the last 10 years as my most senior postdoc, and Clément and Zuri and Ying are all involved in the memory examples that I showed in the new experiments, and Johanna Bergmann with the imagery versus illusion experiment. And I thank you for your attention. I'm happy to take questions.